Professor André, thank you for joining me. Tell me, why did you and your colleagues create this conference?
So, as you know, there is now a lot of initiatives to personalize cancer therapy according to the genomic alteration. And there are two main reasons for creating this conference. First, the patients are being offered some genomic tests that are now available, and there is no education for the oncologist to interpret the result of these tests. So the first reason why we wanted to create this conference was to educate and share some way of interpreting the genomic data from the patients. The second reason why we have created this conference is to have some consensus about how to interpret the genomic alteration. Example, there are different trials across the world trying to show the clinical utility of doing genomic testing, but each of these different trials use a different interpretation tool of the genomic data. So one of the goals is to standardize and to better understand how can we interpret the genomic data.
Precisely, what can we learn from recent trials, and how are these findings going to be reflected in the conference program?
So in terms of what did we learn from clinical trials? So first, there are two very different kind of trials. The first kind of trial are what we call stratified medicine, where we develop a drug in a group of patients who present the same genomic alteration. So this is the first kind of trial. From these trials, what we learn is that there are some genomic alterations that predict efficacy of targeted therapy. Example, EGFR inhibitor, ALK inhibitor, or endocrine therapy in breast cancer. So this is what we call stratified medicine. And this applies for the most frequent genomic alteration. And then we have another kind of trials that are the trials testing high throughput genomics, meaning a large number of genes. And these trials are now ongoing and aim at showing the clinical utility of doing large number of genes in each patient. Until now, what we learned from this trial is mainly feasibility, and we also learned that we need better tools to interpret the genomic data, and this is precisely why we are doing this conference, is to interpret the data from high throughput genomics.
Mm-hmm. How long will it be before personalised molecular medicine and genomic screening will be standard in identifying cancer treatments?
So in some way, personalized therapy is already used in cancer. For example, we give ALK inhibitors according to ALK translocation, or we give HER2 inhibitors when the patient presents HER2 amplification. So what we are preparing now is the next step, is to what extent using or screening for several hundred genes can improve the outcome. And for this, the trials are ongoing, but we are facing some issues that are the fact that we don't know really how to rank each genomic alteration when we screen large number of genes. So what are the next steps in this field? The next steps are clearly to better rank each genomic alteration and to better understand the clinical meaning of each genomic alteration. When we will be able to do that, then we will develop a second generation of trials that really will show clinical utility of this kind of approach.
In terms of current practice, what are the limits of the kinds of techniques being used in cancer treatment?
So in terms of current limitations of the technology, so right now we are using mainly DNA to identify a genomic alteration. There are several reasons for that. First, because DNA is easier to be analyzed. Second is because most of the drivers have been reported, like mutation or gene amplification. But in fact, we already know that this approach is not optimal, because sometimes some mutation do not lead to activation of the pathway, and at the opposite, there are some cancers who do not present mutation but obviously have a progressive disease. So what we think are the next step? The next step is to come with some technology that could really assess which pathway is activated in each cancer, and this can be done using a protein analysis, and we will have one presentation about new proteomic analysis to personalized therapy. This can also be done using gene expression, and we also need some better tool to understand the environment of the cancer. Example, what is the impact of stromal cell, immune cells, but also can we quantify intratumor heterogeneity. So I think the key message is that right now we are doing personalized therapy using DNA-based approach, but the next step will be to use protein-based tools or gene expression array, and to better assess the tumor as a whole.
So then finally, given what we expect from emerging biotechnology, could we imagine that one day cancer diagnosis might become a routine practice with a simple blood test?
I think in the future, blood tests will be part of the molecular analysis, and we will have some presentation during the MAP conference about how to use blood tests and what are the perspectives. But personally, I think it's unlikely that blood tests will summarize the whole portrait of the tumor for many reasons. First, because until now, it's very difficult to assess protein activation using a blood test. Second, is because we know that we need to personalize therapy according to the activation of immune system. And until now, most of the predictors are being developed in the tumor, so we need to assess the immune system in the tumor. And also because the next generation of biotechnology, like metabolomics or other things like this, will be applied in the tumor. So personally, I think that blood tests will be very important tool, but it's going to be only one component of the whole portrait that will include biopsy of the cancer.
Thank you.